I wanted to give you an update on vaccines, which I think is the good news story from today. So there's been a lot of progress in vaccine development, which I'm sure you would have heard in the news. And you can see here the increase in the number of vaccines that are progressing through different phases of clinical trials. So you would have all heard in the news that there's been some announcements from the companies themselves about the success of their vaccines. First of all, the viral vectored vaccines, the adenovirus-based vectors, and I think I described this last time as a Trojan horse. These are pretty innocuous viruses that are used to deliver the code for the spike protein, and that's delivered intramuscularly, and the cells then produce the encoded protein. So the cells produce the spike protein and they generate both antibody and cellular responses. The other two companies that have released data are RNA-based vaccines. And this is a really new technology that I'll just refresh your minds about here. So normally our cells encode DNA. That DNA is copied into what's called messenger RNA. And this is really what we call the data carrier of code. So the messenger RNA codes for all of the proteins that make up our bodies. But you can also use messenger RNA to deliver any sequence that encodes a protein. And in this case, companies have been using the spike messenger RNA of the coronavirus, and that's delivered into cells, and the cells then produce the proteins that generates the antibody and the cellular responses. So the first announcement came from Pfizer in conjunction with BioNTech. This is a 2-dose vaccine schedule, 21 days apart. Each dose contains 30 micrograms, and that's a messenger RNA formulated in a lipid nanoparticle. The phase 1 studies have been published. The phase 3 was a double-blind placebo-controlled trial in various age groups in different, in racially and ethnically diverse peoples. So the results show that the vaccine is 95% effective. They had 9 cases of severe disease in the placebo group, and they did report 1 severe case in the vaccine group. But as you can see, there's a massive difference in the number of cases in the placebo group compared to the vaccine group. So the company is able to supply 50 million doses this year and 1.3 billion doses next year. There are storage issues with this vaccine. It does need to be kept long-term at -70 degrees, but it is stable for 5 days at 4 degrees. And this is likely to be sufficient for pretty widespread use in GP clinics, etc. in the UK, because they have announced that they will begin vaccinating in the UK on Monday with this vaccine. The cost is about $20 a dose, so it's not cheap, but for high-income countries, it is an affordable, fantastic option. The second company that announced their results is the Moderna mRNA vaccine. This is a slightly higher dose, 100 micrograms, 2 doses, 21 days apart. The trial enrolled 30,000 participants. There were 90 cases in the placebo group and 5 in the vaccine group. And what's really encouraging, and you know, this is just a numbers game, these may not be statistically significant differences between the Pfizer and the Moderna trial. In the Moderna trial, they had 30 severe cases, 1 person in fact died. In the vaccine group, they did not have any severe cases. So it does suggest that the vaccines will also reduce the severity of infection if you are unfortunate enough to be infected. The cost for this vaccine is predicted to be $32 to $37 per dose, so it's slightly more expensive. This is a new company. They've never had a product to market, so this will be, in fact, their first product they've taken to market. So they're a little bit slower on the manufacturing side. They can produce 20 million doses this year, but they estimate that next year they'll have up to 1 billion doses of this vaccine available. Australia will be able to receive 0.8 million doses through the COVAX agreement. The other vaccines that have been announced are adenovirus-based vaccines. So we have the Gamaleya Research Institute, which is making the Sputnik 5 vaccine in Russia. This is a human adenovirus vaccine, and they use 2 different serotypes of adenovirus to deliver the vaccine 3 weeks apart. It encodes the spike protein. It has much more favourable storage conditions. So 2 to 4 degrees is its storage. It's a lyophilized vaccine, and it's much cheaper, less than $10 a dose. And there have been 1.2 billion doses of this vaccine requested around the world. In their trial, in the 10,000 placebo group, they had 19 cases. In the 30,000 recipients, they've reported 1 case. Now, these were interim results. These are very small numbers. I think it was a bit of a, we want to announce our vaccine trial results as well a little bit early so that we can say that it is 95% effective, and we still await the full results. The fourth vaccine is the Astrazeneca Oxford University vaccine. This is based on chimpanzee adenovirus. It encodes the spike protein, and it is what we call a homologous prime boost. So the same serotype of adenovirus is used in the prime and the boost. They can produce up to 3 billion doses in 2021. And in fact, Australia is manufacturing 30 million doses at the CSL plant in Broadmeadows, and we will secure another 3.8 million doses from overseas. They have 60,000 people enrolled in trials all around the world, and the cost of this vaccine is about $3 to $4 a dose. Again, only interim results, and this has been a little bit controversial because they had a bit of a mess up with their dosing. So 2,741 people had this, what they call a half prime and a full boost, and 8,895 people had a full dose in both the prime and the boost, and they reported 131 cases. Overall, they report 90% efficacy when they use the half full regimen versus 62% in the full. And I think this is likely to be one of the caveats around adenovirus-based vaccines, where you make antibodies to the actual adenovirus, and this then restricts your ability to boost your antibody response because you've already made antibodies to the adenovirus itself. So perhaps that half prime reduces those antibodies to the adenovirus and allows a proper boost to occur, but they actually need to restart that trial, but they're hoping that this data will be enough to get them emergency use coverage. So what does all of this mean? It's fantastic news for vaccines, but they are only, in a lot of cases, interim results. The data hasn't been peer reviewed. And remember, efficacy is determined by monitoring participants for symptomatic infection confirmed with PCR diagnosis. So although we are getting some information on whether people have had severe disease, it is still possible that you have asymptomatic infection, although I think at such high efficacy rates, this is likely to be not going to contribute significantly to transmission, but we'll have to wait and see. So it's like all of these companies have applied for emergency use for their vaccine. If they get approval, it could be that placebo groups will no longer be allowed in trials because of ethical concerns. The questions that remain is, will the immunity be long lasting? How will it work against the evolving strains of the virus? Some of the companies have tested the immune serum for cross-neutralization against other strains, and they report good neutralization, so no effect on no difference in evolving strains. And we don't know yet how well the vaccines will work in children and pregnant women and other groups of individuals who are not currently covered in the trials. And we don't know, as I said, whether immunity will be truly sterilising, although I suspect at 95% efficacy, it's likely to be pretty close. So there are some implementation limitations, as I said, with the Pfizer vaccine, but I think this can be overcome if it is stable for 5 to 8 days. And we have secured Australia has secured 10 million doses of the Pfizer vaccine, which means when we do approve this, we can begin vaccinating in the new year. So I just wanted to finish on what's so significant about mRNA vaccines. So they have been around for a while, but the breakthrough really came with this discovery in 2005. You can use mRNA vaccines to encode any protein to generate an immune response. And it's very easy to make large amounts of mRNA. The sticking point is the inclusion of the nanoparticle formulation to protect the mRNA. You can generate both an antibody and a cellular response. And These vaccines, and this is really what this discovery was about, have been engineered to minimize the innate immune response. Normally we would see RNA like this as foreign, but it's actually been engineered to avoid that response and also to increase its stability. It means that rapid vaccine development pathways are possible. You can precisely control the type of immune response you want. You can boost immune responses. And in my opinion, this is the most exciting discovery in vaccine technology. And we will be able to make many more vaccines to human pathogens using this technology, and it will get a Nobel Prize in the future. Finally, should we worry about safety? So I know a lot of people have been asking me about safety of vaccines, and we do think this has been a very rapid vaccine development pathway, but it's also related to incidents. So if I compare this to the RV144 trial, which started in 2003 and enrolled 16,500 people, they had to monitor their enrolled subjects until 2006 to actually get a sufficient number of cases to determine whether they had an efficacious vaccine, because the incidence was so low, even in Thailand. And they didn't announce efficacy until 2009. We are doing these vaccine trials in a pandemic where the incidence is extremely high. So in fact, we can enroll people very quickly and get the efficacy data very quickly. The other thing that's important to remember is that we are enrolling people in many, many different trials around the world. In the end, we may have more than 1 million people enrolled in trials. So we'll actually have an enormous amount of safety data, more than we've ever had for any other vaccine that's gone into humans in the past. And in fact, I think a lot of people don't actually understand what the safety concerns were with this vaccine. What we were really worried about was antibody-dependent enhancement. And this is a process where the antibodies that you make to the vaccine actually give the virus another way of entering your cells that enhance infection and cause a massive inflammatory response. And this has been observed for respiratory syncytial virus, dengue, and the SARS coronavirus 1 vaccine candidates. Now, none of the virus challenge studies that have been conducted in macaques for the SARS coronavirus 2 vaccine or any of the human trials have shown any evidence of ADE occurring. So in fact, virologists and immunologists are extremely relieved to see that none of the vaccines have reported this adverse event. So in fact, I think we are really in fantastic shape to be confident of the safety of this vaccine and also the efficacy. Thanks very much for listening.